DNA structure and sequencing. The building blocks of DNA are nucleotides. The important components of the nucleotide are a nitrogenous base, deoxyribose, 5-carbon sugar, and a phosphate group. Figure 1. The nucleotide is named depending on the nitrogenous base. The nitrogenous base can be a purine such as adenine, A, and guanine, G, or a pyrimidine such as cytosine, C, and thymine, T. The nucleotides combine with each other by covalent bonds known as phosphodiester bonds or linkages. The purines have a double ring structure with a 6-membered ring fused to a 5-membered ring. Pyrimidines are smaller in size. They have a single 6-membered ring structure. The carbon atoms of the 5-carbon sugar are numbered 1 foot, 2 feet, 3 feet, 4 feet, and 5 feet. 1 foot is read as 1 prime. The phosphate residue is attached to the hydroxyl group of the 5' carbon of 1 sugar of 1 nucleotide and the hydroxyl group of the 3' carbon of the sugar of the next nucleotide, thereby forming a 5'-3 phosphodiester bond. In the 1950s, Francis Crick and James Watson worked together to determine the structure of DNA at the University of Cambridge, England. Other scientists like Linus Pauling and Maurice Wilkins were also actively exploring this field. Pauling had discovered the secondary structure of proteins using X-ray crystallography. In Wilkins' lab, researcher Rosalind Franklin was using X-ray diffraction methods to understand the structure of DNA. Watson and Crick were able to piece together the puzzle of the DNA molecule on the basis of Franklin's data because Crick had also studied X-ray diffraction. Figure 2. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine. Unfortunately, by then Franklin had died, and Nobel Prizes are not awarded posthumously. Watson and Crick proposed that DNA is made up of 2 strands that are twisted around each other to form a right-handed helix. Base pairing takes place between a purine and pyrimidine, namely, A pairs with T and G pairs with C. Adenine and thymine are complementary base pairs, and cytosine and guanine are also complementary base pairs. The base pairs are stabilized by hydrogen bonds. Adenine and thymine form 2 hydrogen bonds and cytosine and guanine form 3 hydrogen bonds. The 2 strands are anti-parallel in nature. That is, the 3' end of one strand faces the 5' end of the other strand. The sugar and phosphate of the nucleotides form the backbone of the structure, whereas the nitrogenous bases are stacked inside. Each base pair is separated from the other base pair by a distance of 0.34 nanometers, and each turn of the helix measures 3.4 nanometers. Therefore, 10 base pairs are present per turn of the helix. The diameter of the DNA double helix is 2 nanometers, and it is uniform throughout. Only the pairing between a purine and pyrimidine can explain the uniform diameter. The twisting of the 2 strands around each other results in the formation of uniformly spaced major and minor grooves. Figure 3. DNA sequencing techniques. Until the 1990s, the sequencing of DNA, reading the sequence of DNA, was a relatively expensive and long process. Using radiolabeled nucleotides also compounded the problem through safety concerns. With currently available technology and automated machines, the process is cheap, safer, and can be completed in a matter of hours. Fred Sanger developed the sequencing method used for the Human Genome Sequencing Project, which is widely used today. Figure 4. The method is known as the dideoxy chain termination method. The sequencing method is based on the use of chain terminators, the dideoxynucleotides, DDNTPs, the dideoxynucleotides, or DDNTPSs, differ from the deoxynucleotides by the lack of a free 3'O group on the 5-carbon sugar. If a ddNTP is added to a growing DNA strand, the chain is not extended any further because the free 3'O group needed to add another nucleotide is not available. By using a predetermined ratio of deoxyribonucleotides to dideoxynucleotides, it is possible to generate DNA fragments of different sizes. The DNA sample to be sequenced is denatured or separated into 2 strands by heating it to high temperatures. The DNA is divided into 4 tubes in which a primer, DNA polymerase, and all 4 nucleotides, A, T, G, and C, are added. In addition to each of the 4 tubes, limited quantities of one of the 4 dideoxynucleotides are added to each tube respectively. The tubes are labeled as A, T, G, and C according to the ddNTP added. For detection purposes, each of the 4 dideoxynucleotides carries a different fluorescent label. Chain elongation continues until a fluorescent dideoxynucleotide is incorporated, after which no further elongation takes place. After the reaction is over, electrophoresis is performed. Even a difference in length of a single base can be detected. The sequence is read from a laser scanner. For his work on DNA sequencing, Sanger received a Nobel Prize in Chemistry in 1980. Gel electrophoresis is a technique used to separate DNA fragments of different sizes. Usually the gel is made of a chemical called agarose. Agarose powder is added to a buffer and heated. After cooling, the gel solution is poured into a casting tray. Once the gel has solidified, the DNA is loaded on the gel and electric current is applied. The DNA has a net negative charge and moves from the negative electrode toward the positive electrode. The electric current is applied for sufficient time to let the DNA separate according to size. The smallest fragments will be farthest from the well, where the DNA was loaded, and the heavier molecular weight fragments will be closest to the well. Once the DNA is separated, the gel is stained with a DNA-specific dye for viewing it. Figure 5. Evolution connection. Neanderthal genome. How are we related? The first draft sequence of the Neanderthal genome was recently published by Richard E. Green et al. In 2010. Neanderthals are the closest ancestors of present-day humans. They were known to have lived in Europe and Western Asia before they disappeared from fossil records approximately 30,000 years ago. Green's team studied almost 40,000-year-old fossil remains that were selected from sites across the world. Extremely sophisticated means of sample preparation and DNA sequencing were employed because of the fragile nature of the bones and heavy microbial contamination. In their study, the scientists were able to sequence some 4 billion base pairs. The Neanderthal sequence was compared with that of present-day humans from across the world. After comparing the sequences, the researchers found that the Neanderthal genome had 2 to 3% greater similarity to people living outside Africa than to people in Africa. While current theories have suggested that all present-day humans can be traced to a small ancestral population in Africa, the data from the Neanderthal genome may contradict this view. Green and his colleagues also discovered DNA segments among people in Europe and Asia that are more similar to Neanderthal sequences than to other contemporary human sequences. Another interesting observation was that Neanderthals are as closely related to people from Papua New Guinea as to those from China or France. This is surprising because Neanderthal fossil remains have been located only in Europe and West Asia. Most likely, genetic exchange took place between Neanderthals and modern humans as modern humans emerged out of Africa. Before the divergence of Europeans, East Asians, and Papua New Guineans, several genes seem to have undergone changes from Neanderthals during the evolution of present day Humans. These genes are involved in cranial structure, metabolism, skin morphology, and cognitive development. One of the genes that is of particular interest is RUNX2, which is different in modern-day humans and Neanderthals. This gene is responsible for the prominent frontal bone, bell-shaped ribcage, and dental differences seen in Neanderthals. It is speculated that an evolutionary change in RUNX2 was important in the origin of modern-day humans, and this affected the cranium and the upper body. DNA packaging in cells. When comparing prokaryotic cells to eukaryotic cells, prokaryotes are much simpler than eukaryotes in many of their features. Figure 6. Most prokaryotes contain a single, circular chromosome that is found in an area of the cytoplasm called the nucleoid. The size of the genome in one of the most well-studied prokaryotes, E. coli, is 4.6 million base pairs, approximately 1.1 millimeters, if cut and stretched out. So how does this fit inside a small bacterial cell? The DNA is twisted by what is known as supercoiling. Supercoiling means that DNA is either under wound, less than 1 turn of the helix per 10 base pairs, or over wound, more than 1 turn per 10 base pairs. From its normal relaxed state, some proteins are known to be involved in the supercoiling. Other proteins and enzymes such as DNA gyrase help in maintaining the supercoiled structure. Eukaryotes, whose chromosomes each consist of a linear DNA molecule, employ a different type of packing strategy to fit their DNA inside the nucleus. Figure 7. At the most basic level, DNA is wrapped around proteins known as histones to form structures called nucleosomes. The histones are evolutionarily conserved proteins that are rich in basic amino acids and form an octamer. The DNA, which is negatively charged because of the phosphate groups, is wrapped tightly around the histone core. This nucleosome is linked to the next one with the help of a linker DNA. This is also known as the beads on a string structure. This is further compacted into a 30 nanometers fiber, which is the diameter of the structure. At the metaphase stage, the chromosomes are at their most compact, are approximately 700 nanometers in width, and are found in association with scaffold proteins. In interphase, eukaryotic chromosomes have 2 distinct regions that can be distinguished by staining. The tightly packaged region is known as heterochromatin, and the less dense region is known as euchromatin. Heterochromatin usually contains genes that are not expressed, and is found in the regions of the centromere and telomeres. The euchromatin usually contains genes that are transcribed, with DNA packaged around nucleosomes but not further compacted. Section summary. The currently accepted model of the double helix structure of DNA was proposed by Watson and Crick. Some of the salient features are that the 2 strands that make up the double helix are complementary and anti-parallel in nature. Deoxyribose sugars and phosphates form the backbone of the structure, and the nitrogenous bases are stacked inside. The diameter of the double helix, 2 nanometers, is uniform throughout. A purine always pairs with a pyrimidine. A pairs with T, and G pairs with C. 1 turn of the helix has 10 base pairs. During cell division, each daughter cell receives a copy of the DNA by a process known as DNA replication. Prokaryotes are much simpler than eukaryotes in many of their features. Most prokaryotes contain a single, circular chromosome. In general, eukaryotic chromosomes contain a linear DNA molecule packaged into nucleosomes, and have 2 distinct regions that can be distinguished by staining, reflecting different states of packaging and compaction. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.